Hello, this is Tommy. Welcome to Chatomics. So today I'm going to introduce you a very cool R package called ggside. So ggside is an R package that provides a new way to visualize data by combining the flexibility of ggplot2 with the power of side-by-side plots. So to demonstrate, we'll use a single cell data set. So I have a link here, so you can go there and to read the tutorial. So if you scroll down to the end, so this is the type of figure you are able to make using ggside. So the main plot is a scatter plot. Then you can have either box plot on top or density plot on the y-axis. Okay, let's go back to the studio. Okay, so first, if you haven't installed this package, use install.package to install it. And I have already installed it, so I will just load those packages. Because I'm going to use the thread object to demonstrate this, I'm going to load the thread data object library as well. Okay, once you load this thread data library, so you run this function data PBMC 3K, and this PBMC 3K thread object will be available for you to use. So this is just 3,000 immune cells from blood and single cell sequenced by single cell technology. Actually 2,700 cells. And we will just do some routine processing here for this thread object. So normalize data, find the most variable genes, scale the data, run PCA, find neighbors, and find clusters, and run UMAP. So those are just routine processing steps for single cell data. So if you are not familiar with single cell RNA-seq processing, I recommend you to read the tutorial from Thread website. Okay, this thread object comes with the annotation for each cell. So I just use this function idents PBMC3K to assign each cell the idents for with the annotations. Okay, let's just run it. And now let's make a dim plot. Oh, by the way, I haven't run this pre-processing step yet, so that's why the UMAP is not there. I think it will take a couple of seconds. It's a relatively small data set. But remember, in a real data set, you can have like millions of cells. It can take quite long to run even those pre-processing steps. Okay, it's done. And let's make the dim plot. It's essentially just a scatter plot. Let's rerun the previous line to make each cell's annotation with the cell types. Okay, let's rerun it again. So now here is just the cluster ID. So we want the cell label here. So let's do this again. And you see different cell types, they kind of separate in this two-dimensional UMAP plot. So you have B cells here, dendritic cells, platelets, CD14 positive monocytes, and this is just CD16 positive monocytes. And you have the T cells in here and NK cells. Okay, to demonstrate this ggside package, I will actually have some helper functions to extract the gene expression value from the thread object. So by the way, I have this R markdown in the description of this video. Download it and you can follow the tutorial. So let me just run this. And this is just a helper function to get the gene expression in a data frame format. So for example, if I run this and provide a thread object name and the genes that you want to, the values you want to extract. So if you just run that, and we'll combine all this metadata columns in the metadata slot from the thread. So look at that. So essentially you add these two columns, like those two genes, and this is the value of the gene expression. And the rest are just the metadata columns from the thread object. Okay, So with this data frame, so now you can plot a plain scatter plot. So let's focus on here, CD14 positive monocytes and CD16 positive monocytes. So that's why I filter the thread annotation, essentially this column, to be only those two cell types. And then make a scatter plot. X-axis is the CD14 expression, and Y-axis is the CD14 expression. So CD14 is also FCGR3A. And let's see how it looks like. Okay, now it's just a plain scatter plot. So, and you see those are the CD14 positive monocytes, and you see, okay, those are CD14 positive, but CD16 negative. So those are the CD16 positive monocytes, so that they are CD16 positive, but CD14 negative. But you also see some of those cells, like they're in the maybe intermediate state, so they have both a CD14 and CD16 expression level. Okay, so what we can do here is to add two box plots, one on the x-axis and the other one is on the y-axis for the expression value of CD14 and CD16. So to do this, so you'll use this function called ggside boxplot and ggside boxplot from the ggside package. Okay, the rest is the same, and let's run it. The rest of the code is the same as here, scatterplot. So let's run it. So now, like you see nicely, ggside add these two box plots on the two sides of this scatterplot. And you see here for CD14, and you have high expression of CD14 in CD14 positive monocytes. And here for CD16, FCGR3A, and you have higher expression for CD16 positive monocytes, which makes sense. But of course, you also have those like outliers here, which demonstrate those actual intermediate maybe cells. Okay, that's pretty nice. So ggside, you can also add a density plot. For example, if you just change one of them to be the geome Y density, and let's see how it looks like. Now you have actually one box plot here and one density plot here, so which is pretty actually informative. Okay, that's the way you use ggside to make this side by side plots. But we can also use another R package called patchwork. So patchwork is a package to make composite ggplot figures. So if this is the link, and it actually gives you really powerful ways to combine different ggplot figures. So for example, if you have two ggplot objects, P1, P2, you can just use the plot sign to make them together. Okay, so I highly recommend you to actually read the tutorials and also guides here. Okay, let's go back here. So let's actually recreate a similar plot using patchwork. But to do this, actually, we're going to actually assign this as a P1, and there's this empty space here. We can use this called plot spacer function to denote empty space, and this is the P3 and this P4. No, this P2 and this P3. So we have P1, P2, P3, and this one is just the empty space. Okay. So, okay, let's assign this to P1. So here essentially just a regular box plot. If I run it, you will see it. It's just a regular box plot. But if I do chord flip, you actually flip it like this. So we assign this to P1. Okay. Oh, P2 essentially just the scatter plot, right? But we remove the legend by using theme legend position dot position equals to none. Okay, so this is P2. And P3 is the other box plot. So let's do it. P3 and P3 is just the other box plot. Okay, now we can combine all those three plots together, including this empty space here. P1 plus this empty space, P2, P3, and you specify the layout by using plot underscore layout function, and you can specify the width of those two columns because now you have 4 figures, 1, 2, 3, 4, and this is just width of those two columns, and this is the heights of those two rows. So you can specify different ratio, and you specify guys equals collect. So all if they are of the same actually legend, so it will be combined into a single one. So let's see how it looks like. Oh, so I think I have to load the patchwork function library first. Okay, let's see. Oh, P2 is not available. Okay, we haven't run this line of code yet. Okay. Okay, now let's run it. See, it's a very similar actual plot to the one that was produced by using the ggside, but it's much actually easier. But then patchwork is much more flexible. So I hope you enjoyed this video. Make sure you click subscribe if you like this content. See you next time.